Hello students, so welcome back to the channel. In this video, we are going to study about light reaction, cyclic and non-cyclic photophosphorylation. Now see what are the steps which light reaction include. First of all, photoactivation of the chlorophyll will be done, then splitting of water molecule, along with its oxygen release and formation of high energy chemicals like ATP and NADPH. Now before we study how the chlorophyll is working in the chloroplast, let us see what happened when any pigment molecule absorbs the light. Now, basic concept, understand it. Whenever any light is absorbed, that color will be disappeared from the transmitted or reflected light. But energy has not disappeared. So what happened whenever any molecule absorbs a photon of light, one of the electron of that molecule is elevated to a higher energy level. And we say now the pigment molecule is excited. After moving the electron away from the atomic nucleus, when we say pigment molecule is excited, the excited state is actually unstable. So generally, when isolated pigment, they absorb light, their excited electron come down back to the ground state, releasing excess energy in the form of a heat. But when the chlorophyll in isolation, chlorophyll will emit not only heat, it will emit light also, which is known as fluorescence. You can see here how isolated chloroplast, when the electron come back from the excited state to the ground state, not only heat is liberated, excess is liberated in the form of fluorescence also. It is in the isolated. But in the plants, chloroplast is the area where chlorophyll are present. Is it clear? So they are not in the isolation. Chlorophyll pigment molecules and other accessory pigments, they are organized in one special photosystems. Okay, we have already discussed in the previous video. If you want, you can refer it also. Two type of photosystems are there, photosystem 1 and 2, and they differ with respect to the chlorophyll molecule whose reaction center is different. In the PS1, reaction center is of 700 nanometer, and in PS2, reaction center is of 680 nanometer. So chloroplast is a chemical factories which is powered by the sun. They are thylakoids. They will transform light energy into ATP and NADPH. So chlorophyll molecules, after absorbing the light, produce very different result in the chloroplast, which what they do in isolation. Okay, that is quite different from the isolation. Here result is different. Why? What is the reason why the result is different? Because of their environment, the location, okay, and other molecules with which they are associated. Because of this, the result is different. What happened here, if you see, the light energy not only help to boost their electrons to the higher energy level, but also provide energy to transfer it to different molecule. That is primary electron acceptor. See here, this is your accessory pigments, light harvesting complex, already explained in the previous video, please check it. This is chlorophyll molecule. This is one chlorophyll molecule according to our NCERT. This is in the reaction center, and this is primary electron acceptor, reaction center. So this arrangement is in such a way that electron not only excite to high energy level, but it is also given to primary electron acceptor. And the pigments are organized to form photosystem. So this is the first step of light reaction. The first step of light reaction is photoactivation of chlorophyll. And it is a redox reaction. Now what will happen? Once the electron is moved from the photosystem 2, that electron should be replaced. Now after losing the electron, the chlorophyll, which is present in the reaction center of PS2, work as a powerful oxidizing agent, and it causes the water molecule near to split. Is it clear? It means splitting. Who is doing? PS2 will do the splitting of water molecule. See the equation here. When the water molecule is split, oxygen is a product which is released, and hydrogen will break into protons and electrons. So electrons, they are getting it, and protons, they will accumulate in the lumen. Okay, they are releasing the proton in the thylakoid space. Now, electron from primary electron acceptor will be given to the photosystem 1 and through electron transport system. Now see electron transport system. In PS2, we know reaction center has chlorophyll A, absorption 680. Electron is excited, jumps into the orbit away from nucleus, and it is picked by the electron acceptor. Now from primary electron acceptor, it is going from PS2 to PS1. And the chain through which it is passing, that is known as electron transport chain, which generally consists of many pigments, but main pigment is cytochromes. These are the proteins, actually, these are the proteins which are having iron content. So they are the cytochromes. So here several protein complexes are involved, but main is cytochrome. So this movement of electron is again downhill in terms of oxidation reduction. In the meantime, the electron from the reaction center of PS1 is also excited after receiving 700 nanometer. It will give it to another acceptor. And here again, the electron will move downhill. It will come to the NADP plus, and NADP plus will get reduced to NADPH. So this is an analogy to explain the linear flow of electrons. See here, this is photosystem 2. When the light is striking, the electron is going accepted by primary electron acceptor, coming downhill, okay? And during this time, only ATP formation will be done. Then it is going to PS1. PS1, again, it is losing the electron accepted by primary electron acceptor of PS1, and finally to NADPH. So this is just an analogy to explain you how the linear flow of electron is working. See here in this diagram also, you can check with it. This is the way how the electron flow is going on. Now, during this electron flow, we are able to make ATP. Plants are able to make ATP. The process by which they make ATP, that is known as phosphorylation. So they are having the capability to extract energy from any oxidizing substance and store this in form of bond. So ATP is the one which they prepare. The ATP is made by chloroplast and mitochondria, and the process is known as phosphorylation. But right now we are discussing in the chloroplast, so we will say photophosphorylation. Why? Because here the light energy is used to prepare ATP. It is synthesis of ATP from ADP plus inorganic phosphate in the presence of light. So two type of, depending upon the electron flow, in this case, if you see two photosystems are used, we will say non-cyclic because it starts from photosystem 2 through electron transport chain. It is given to photosystem 1 and NADP finally. Okay, that previous we studied right now. So both the photosystems are connected through electron transport. It is a Z scheme because of the shape, and both ATP and NADPH are made here. So in this case, electron which start its journey from PS2 never come back to PS2. It is finally given to NADP plus. But in case of cyclic, it starts only from PS1, come back to PS1. So when only PS1 is functional, electron is circulated within the photosystem and phosphorylation occurs because of the cyclic flow of electrons. And where this possible location is, it is in stroma lamella. Stroma lamella, why? Because that is the area which is non-oppressed. Clear with a non-oppressed. Please see the previous video to see what is oppressed region of grana and what is non-oppressed. So stroma lamella is non-oppressed, so it does not have PS2, it does not have NADP reductase. So electron is not able to go to NADP plus, it is coming back to PS1. So it results only in the synthesis of ATP and not NADPH. So I hope so in this video, today what we discuss about light reaction, photoactivation of chlorophyll, then photolysis of water, and finally the electron will be transported in the electron transport chain from PS2 to PS1 and finally to the NADP plus. Z scheme, and in this case it is non-cyclic. NADPH is made plus ATP is made. So it's a non-cyclic photophosphorylation. But when only PS1 is functional, then only ATP is made, NADPH is not made. So I hope so in this video, this topic might be easy for you. In the next video, we are going to study about chemiosmosis. Thanks for watching.